Hello everybody. I'm very happy to have Professor José-Alain Sahel with us. Dr. Sahel is the chair and the distinguished professor of the Department of Ophthalmology at the University of Pittsburgh at the School of Medicine. He's the director of the UPMC Eye Center and the Eye and Ear Foundation Endowed Chair of Ophthalmology. Dr. Sahel studied medicine at the University of Denis Diderot at Paris and ophthalmology in the University of Louis Pasteur at Strasbourg. He founded and directed the Vision Institute in Paris from 2008 to 2020, and is currently an exceptional class professor at the Sorbonne Medical School and hold many other positions in the field of ophthalmology and neuroscience. He is very known worldwide for his expertise in vision restoration technique. He has developed several interventions, including stem cell implantation, gene therapy, innovative pharmacological approaches, and retinal prosthesis, all for the treatment of retinitis pigmentosa and other retinal dystrophies and age-related macular degeneration. Over the past decades, he had led pioneering effort in optogenetic vision restoration, and we have heard already part of this. He has published more than 600 peer-reviewed papers, and is inventor of more than 40 patents, several of which have already led to a startup companies, including Fovea Pharmaceutical and Sanofi Aventis, GeneSight Technologies, and Pixum Vision for retinal prosthesis, as well as other companies. He is a member of 11 editorial boards. We have several positions in the Institute of Ophthalmology in London and in Strasbourg, and he led many dozens of clinical trials. He got numerous honors, which include Officer of the Legion of Honors and the National Order of Merit, and is invited professor in several institutes. So I'm very happy to welcome you, Professor Sahel, and to here we are looking very heavily to have you with us and to hear your talk. Thank you very much.
Thank you, Yossi. Thank you for the invitation. As I told you, because you already heard from Botan Roska and Serge Picot about the optogenetic part of my research, I decided that I would not bore you again with that and speak about a long strain of research that we conducted over the past several years on gene therapy for Leber's optic neuropathy that has now led to several clinical trials. And so it's going to be more on the translational side from the bench to bedside with significant results from the clinical trials that are very promising. So obviously this is the work of many people. And also this has led to a couple of companies. The one that is mostly involved in that is GeneSight Biologics, that is developing this technology. So as a reminder for the few of you that don't know about it, so Leber's optic neuropathy is a mitochondrial disease affecting the optic nerve. The gene mutated is on the respiratory chain. It's inherited from a mother. It's an orphan disease. And what happens is that patients initially have normal vision for 1 decade, 2 decades, sometimes more. And then suddenly the first eye starts to have blurred central vision. And over the course of a few weeks, at the maximum a few months, the fellow eye is going to lose vision. And most of these patients over the course of 1 year end up being legally blind, which means that their visual acuity is off chart. This disease is really a bilateral disease in 97% of cases. A few cases are very bilateral immediately, but most cases are, as I said, with a small delay between the two eyes. The gene involved in the disease is called ND4. It's one of the genes of the respiratory chain in mitochondria. The exact incidence and prevalence is not fully accurate. There are some very good studies from Northern Europe and some very good studies conducted by Patrick Buman in the UK. So the data we have is that the prevalence is around 1 out of 31,000, so not very frequent, but actually we see a significant number of these patients. In parallel to the loss of central vision in these patients, there is a change in the thickness of the nerve layer on the OCT. Initially some swelling at the time of onset, and then a decrease in density at the level of the optic nerve layer in the OCT. So this disease was considered as untreatable, and the only exception being the idebenone that has been tested and had some temporary approval in Europe, but is not yet approved in the US. And actually, just recently, some European countries like Europe decided to stop funding this treatment because the efficacy was considered as not totally obvious, although there is certainly a trend towards efficacy. So many years ago, my main field is retina, but I see also patients with other type of conditions. And many years ago, it was in 2003, I think, I was in a clinic seeing a young patient with sudden loss of vision in one eye, which was undiagnosed at that time. And he was coming from an advice for this loss of vision. And clearly there was some failure of the optic nerve already, but nothing else, change in the visual field in one eye. And I thought it could be Leber's optic neuropathy. And at that time, the time to get the genetic testing was pretty long. So I thought that the diagnosis was very likely. And unfortunately, I also had to tell the family that this kid that was at that time 15, I think, was very likely to develop loss of vision in the fellow eye. Before I said that to the family, I called the neurologist in the hospital. It was at the Rothschild Foundation. And I asked, did I miss something? Is there any treatment that is working? He said, well, idebenone is not proven and the steroids don't work. I know that some groups are working on gene therapy, but so far the results are not very good. So he sent me actually a paper from the group of John Guy, who was working on gene therapy in, I think, in Annals of Neurology. And I had to tell the family that there was nothing we could do. And I read the paper during that night. And clearly the efficiency of gene therapy was very, very low, less than 5% of the transcript getting into mitochondria. So this didn't look like something very promising. So at that time, and for many, many years, and actually up to now, this disease is considered with very low visual prognosis. Most patients remain legally blind. A few patients improve vision over time, but depending on the mutation, for some mutation, there is almost no improvement, less than 10% patients. Some patients, around 30% with different mutation, can get some spontaneous improvement, but not returning to normal vision. The impact of quality of life, employment, education, and obviously depression is very significant. So the day after I met with this patient, I met a scientist, a woman who had been working in the yeast at École Normale Supérieure in Paris on very basic science. And she had been working on mechanisms where the mRNA is targeted to the surface of the energetic factory in the cells. And she had identified that something which is a very important mechanism was of interest. At that time, she was a very basic scientist and she didn't think there was a very strong clinical application, but she was advised to come and meet with me to think about it. So what she was working on is something which is probably worth spending some time because it's at the core of the science we developed later on. The mitochondria have 1,000 proteins. 10 of these are encoded at the level of the DNA in the mitochondria, which is a very specific code, very different from the code that is encoded at the nuclear level. Still DNA, RNA, and everything, but a different one, but only 10 of them. And actually, ND4, the protein that is mutated in Leber's optic neuropathy, is one of these proteins. It was identified by Wallace in the US in the 1990, the first gene in the mitochondria involved in the disease. And this gene is encoded at the mitochondrial level. But then there are 990 other proteins that are present in the mitochondria that are encoded at the nuclear level. And there is a mechanism to target the mRNA that is encoded at nuclear level to the surface of the mitochondria to release the RNA there and to have a protein engineered in the mitochondria. And so to get that, there is a tag that is put on both sides of the RNA that is targeting the RNA to the surface of the mitochondria to express the protein. So it's called mitochondrial targeting sequence, and it's on both sides of the RNA. And so the idea was that if this is what makes this protein express in the mitochondria, maybe to cope with the mutations in the mitochondrial genome, we could use the same tag, transcode the genome at the level of the nucleus, and express it using gene therapy and get it expressed in the mitochondria. So this was the idea. We spoke with this scientist, and I told her, I think I know a disease where this would make a big difference. It's a Leber's optic neuropathy, because as I had read the day before, it was not working at that time. And so we started to join forces. Her name is Marisol Corral-Debrinsky. So the idea is to use the protein import into mitochondria from the RNA at the nuclear level. And the proof of principle was obtained with 4 different proteins, including in ND4. And this was published at the time many times, actually, many papers. The most important one being in 2008 in the American Journal of Ophthalmic Genetics, and showing the efficacy for ND4. So from there, we embarked into a journey to try to cope with the disease in the LH1, Leber's optic neuropathy. So the mitochondria are very important for axonal transport in these optic nerve. As you know, the nucleus is at the level of the retina, and the axons are forming the optic nerve and are joining at the level of the CGA, and then the second neuron starts from them to go to the cortex. So this inefficiency of mitochondria due to the mutation, we don't know exactly why. It's present from birth, and not every patient with a mutation develops the disease. Actually, only 30% of women and only around 50% of men. And this occurs acutely over the course of a few weeks in one eye and then the fellow eye. So the idea was to target the genome in these cells, the RGCs, to get the expression of a normal protein and to compensate for that. So this is what we did. And the idea was to develop a product to really compensate for the mutation that occurs, as I said, at the level of the respiratory chain in the membrane of the mitochondria. And as you see, all the chain occurs there with coenzyme Q, cytochrome C, all of that. But the mutation is in ND4, which is the initial complex, the complex 1 in the respiratory chain in the mitochondria. So this is the mitochondrial genome. And so the idea was to build a vector that would contain the ND4, so this part of the mitochondrial genome, plus the tag sequence that would be put. But in our case, we decided to put it on both sides to get the best targeting. And this was shown to be efficient in the RGCs, and we created a rat model of a disease that was overexpressing the mutant protein, and it was compensated by the normal protein with stable expression and actually vision improvement in the rat. We also tested that in fibroblasts from patients and showed that this was restoring the respiratory chain in these fibroblasts. So this led to the idea to develop a therapy for patients. So the initial thing was to develop it and to demonstrate that in the mutant rat and in the fibroblast. And the gene therapy was developed this way. It was licensed to GenSite that we created at that time. So the mitochondrial genome is transcoded, expressed with a tagging sequence in an AAV, transfecting that in the nucleus. And then the tagging sequence, MTS1 and MTS2, mitochondrial targeting sequence, are targeting that to polysomes at the surface of the mitochondria to get the protein translocated into the mitochondria to restore the energy and the respiratory chain. So from the preclinical work, we did primate studies to work on the pharmacodynamics, to work also on the biodistribution and the safety. And this led to the clinical trial. The initial trial, which is not in my presentation, was a phase 1b/2a. And this trial was done with a dose escalation in 3 patients each, only in Paris. The PI was Catherine Vignal at the Rothschild Foundation, a very good neuro-ophthalmologist in Paris. And 15 patients were treated with a dose escalation with different stages of the disease, but we started with patients with very low vision. And this dose escalation enabled us to define the best dose and the dose that could be both efficient and non-toxic in patients. And this led to start 2 phase 3 clinical trials. One was called REVERSE, and it was designed to target patients that had the onset of disease on the first eye between 6 months and 1 year. And 37 patients were enrolled in that study. And then RESCUE is a study that was conducted in parallel, and that was for patients with less than 6 months of onset. The good news is that despite the fact that this is a rare disease, we were able to really enroll enough patients in the study. So the study was conducted in Europe, in Paris, London, Munich, and in Parma, with very good experts of LHON, and in the US, in Philadelphia, Atlanta, and Los Angeles. And more recently, a few more centers were added to the next study. So the study was designed as a double mask, multi-center trial. One eye was randomized. It could be the first eye or the second eye, and the second eye was receiving a sham injection. The randomization was balancing for severity of visual impairment. So GS10, the therapy product in one eye, sham in the fellow eye. Same thing for the RESCUE. And so there was one group with a GS10 in one eye, right eye, and one group with GS10 in the fellow eye. The primary endpoint was defined as the difference in visual acuity from baseline between the treated and the sham eye. And the goal, and initially the design was designed for 48 weeks, the goal was to show a difference of 15 letters. This is the classical FDA requirement, or visual acuity above 20/200 in a significant number of patients as compared to the untreated. So this was the study, plus a lot of analysis of OCT, visual field, color vision, contrast vision, and quality of life assessment. So the study was conducted. It took more than a year to enroll the patient and then to do the follow-up. And this is how all the study was developed, from the preclinical studies in the animals, the fibroblasts, the biodistribution in primate, the phase 1b/2a tolerability dose, the 2 studies, the phase 3, the long-term follow-up, and I will get back to that in a short moment, a natural history study that was conducted in parallel, and then another phase 3 trial with bilateral treatment that I will describe in a short moment. And what happened is that we had very significant improvement, actually, in the range, if you compare to the lowest point in visual acuity, more than 28 letters in all the treated eyes. But the big surprise was that the non-treated eye, the sham-treated eyes, also had an improvement. And this was totally unexpected. So this was both in the REVERSE trial and in the RESCUE trial. Between 76% and 71% of patients achieved more than 15 letters from the lowest point, more than 5 lines of visual acuity. Much better, as we'll describe, that as compared to natural history. But the big problem is that the effect was bilateral. The effect has been stable, and I will get back to that over more than 3 years, but we couldn't understand why the fellow eye was improving. So this was viewed as a bad news. It was very good news for patients because, as a reminder, these patients have less than 10% chances or maximum 10% chances of visual improvement, and certainly not 5 lines of visual acuity. But why did the fellow eye improve? Many people say, well, it might be a placebo effect, and what's happening? So we wanted to understand that, and we conducted a mechanistic study in the primate. The goal was to see whether there was some diffusion of a vector from one eye to the fellow eye. And indeed, one eye was treated and the fellow eye was compared. And as you can see, the product was found in the treated animals, in the fellow eye, mostly in the retina, the optic nerve, the optic chiasm, and the tractus. Not up to the visual cortex, but you can find that. There was also some level in the anterior segment. And so this was detected, quantified, and there was clearly a diffusion to the contralateral eye, which we didn't know of. And we would never have designed the clinical trial this way, but this was probably the explanation of a bilateral improvement. And so the likely hypothesis is that the vector is diffusing from the treated eye to a fellow eye through the optic chiasma. David Kaltkins at Vanderbilt has done a lot of studies on exchange between the two optic nerves, and he believes that this is probably through the astrocytes, but we don't know that yet. And most likely there is also some diffusion behind the chiasma to the geniculate and maybe to the cortex, but we don't have a strong evidence for the cortex at 3 months. At 6 months, there was more in the cortex. So because of that, we had to find a way to understand what was happening. So we decided to conduct an indirect comparison with all the published studies with natural history of the disease. And so this was conducted with a couple of analyses of many studies. So between the treated patients, together rescue and reverse, that patients were being followed over the course of 3 years now. It's 76 patients, 152 eyes, and both eyes had an improvement versus the untreated group, which is a control group. We had our own natural history study, plus all the analysis of 10 studies that were found of good quality, 2 prospective and 8 retrospective, with patient matching the age, matching the history of the disease, the mutation, and confirmed a mutation in ND4. So this study was published a couple of weeks ago, and this is the comparison. So this is all the patients treated with rescue reverse, and this is the change in visual acuity from baseline. And this is what happened in the untreated group. And the difference between the untreated and the non-treated group, the natural history study, is more than 16 letters from baseline. It's much more from nadir. And also something very important is the shift between off-chart, so not being able to read any letter, versus being able to read. And as you see, the shift is very significant. Most patients that are untreated end up off-chart, not being able to read anything, whereas all patients in the study, and we have all the time points, had an improvement. And some of them, at least the average was more than 16 letters, and most of them had a very significant improvement. So still disappointing because the fellow eye had an improvement, but actually good news for the patients. So then we had another study that resulted from a discussion with the FDA that was started before we knew about the bilateral effect. And so the FDA suggested the following design. One group of patients would be treated in both eyes, the first eye affected and the second affected eye in parallel within the next day or the next week. And another group of patients would get one treatment in one eye and a sham injection in the fellow eye, placebo. And this study included 98 patients, all of them with vision loss less than 1 year. And it took around 1.5 years to enroll all the patients across 13 centers worldwide. And so 108 study patients were enrolled, 98 patients were randomized with an intention to treat. And despite COVID, almost all patients, because if you look at the date of the enrollment and the date of COVID, you find that this was hitting just at the time of COVID, but there was almost no patient lost from follow-up, only one patient lost despite of COVID, because he couldn't come for all the visits, not because he was not happy with the treatment. And so this study comprised 48 patients that got bilateral treatment, 50 patients that got unilateral treatment. The proportion of male versus female is classical for the disease. The age of onset, as you see, is around 31 years on average. The baseline characteristics are very important to define. The mean of time of onset was 8 months. before treatment. So this is more like the reverse group of patients between 6 months and 12 months. And the visual acuity in the first treated eye, the first affected was around 2800 Snellen, 1.64 logMAR, whereas the second eye, which was more recently affected, had a little bit better visual acuities, 2600, 147 in logMAR. And so the time for treatment for the first eye and for the second eye are quite, the two groups is very similar. I mean, the average is the same, time for treatment on average. And this is the time of follow-up that we have. So we reported at 18 months. Actually, we have more now on follow-up in this study. And so I'm going to spend some time on this graph. So if you normalize visual acuity at baseline and you compare, the first treated eye in the bilateral group had a very significant improvement, 0.23 logMAR, P far below 0.001 versus baseline. We had a significant improvement in the second treated eye, so this is the one in orange, in these patients. And these patients were treated a little bit after, a bit closer to loss of vision. There is an improvement in the first eye of a patient treated unilaterally, but there is no significant improvement in the second eye of a patient treated unilaterally, namely the patient that got the sham injection. There is some improvement, but very limited, around 4 letters. So significant improvement in all the treated eyes and a better improvement in the patient that got a bilateral treatment. When you don't normalize with visual acuity at baseline, then you compare the change from start in the bilaterally treated eyes. This is what you see in red. Bilaterally treated eye, the second eye, this is what you see, and you see they end up with the same visual acuity. And when you compare the visual acuity in the unilateral treatment, they end up with the same visual acuity, but one is starting higher, because as you know, logMAR is inverted as compared to the other. So this is the second treated versus the first treated. And as you can see, there is an improvement with treatment, and they end up in the same ballpark. So all very consistent with the data we had from reverse and rescue, and also showing that the bilateral treatment may have a benefit in these patients. So to summarize the change from baseline, for the bilaterally treated patients, an improvement of 12 letters or 8 letters for the second eye. For the unilaterally treated patient, 8 letters or 4 letters, which is not significant. So, and then if you compare to nadir, the lowest point in visual acuity, you have a very significant improvement in the first treated eye. And you have a significant improvement in the second treated eye when you are comparing to nadir, and even in the placebo group. And as you know, this is due to the bilateral treatment now that I showed you the data from the primate. Then there was an analysis of responders, because do we have like a few patients that are driving the responses of all the groups, which means that it could be only a few patients that have a good response and the others would not be driven. And actually, we have a rate of responders of 68% in the first eye bilateral treatment, 53% in the second eye of bilateral treatment, 43% in the first eye of the unilateral, and 15% in the second eye. So very consistent across the dose. And this is the off chart to on chart from baseline to at 1.5 years. So this means that this patient couldn't read anything before treatment, and this patient shifted to being able to read, and the rate of responders is very high. The way you define a responder from nadir is the change of at least 2 lines. This is the definition that was adopted by an international consortium, and that the EMEA has accepted too. And you see the rate of responders, that is 69% for bilateral, 62% for unilateral. So good rate of responders. This, if you want to define the responders for 0.3 logMAR, so 3 lines from nadir, you have a similar percentage. And again, I showed you the curve already a moment ago. So then if the question is on chart versus off chart, 85%, 72%. So it's not just a few patients driving the response, it's really very consistent across all the groups of patients. A question about safety, it's an intravitreal injection. You may have seen recently data about safety issues with intravitreal injections. So there was no serious, there was no systemic effect. Some patients have intraocular inflammation, but was treated topically. I think one patient required systemic treatment. All patients got around the injection some systemic treatment because many patients, especially in the US, couldn't come back immediately. So to be on the safe side, they got the systemic treatment for a few days around the treatment, but no patient had to stay under that. No patient had elevated IOP that was durable. So a very good safety profile in both unilateral and bilateral groups. So because of the initial effect that were reported, in Europe, there was a temporary approval that is now a cohort approval in France, and 22 patients were treated as a compassionate use because of that. And in Italy, it's also provided to individual patients, 7 patients in Italy. In the US, 18 patients got it for free as an expanded access because the FDA approved it to be given as a compassionate use. Now there was some delay because of the PACTI, the fact that the US have prioritized the COVID vaccine in the manufacturing, but this is coming back. So now the company is filing for approval in Europe and will file in the US despite the small difference between the untreated versus the treated eyes in the same patient. So the summary is that there's a significant improvement in visual acuity in treated eyes. From nadir, it's very significant. There is a contralateral effect that is confirmed in the untreated eye on the same patients, like in all studies. There is a trend for a dose effect. And altogether, the safety in almost 190 patients was very good, plus 47 patients that have been treated with compassionate use already in the world. And the safety profile is comparable between the unilateral versus the bilateral treated patients. So the milestones are filing in the EU and then probably filing in the US next year. Just the list of investigators, several in Italy, in the US, so in New York, in Atlanta, in Los Angeles, sorry, I have to go back, and in Philadelphia, many centers in Europe, and also one center in Taiwan, and a center in the UK too, at Moorfields. So many publications, if you want to look, are already, I think, 12 publications on all these studies. The most important that described the mechanistic effect was published in Science Translational Medicine last year. I'm the corresponding author with Patrick Bowman on that one. Thank you very much.
Okay, so let's all thank our speaker. Thank you. So we have some time for questions. So anyone? So maybe I will begin. So first of all, it was very exciting to see that there is so much to do today with genetic treatment and several approaches. And I'm still very excited about the bilateral effect. And was this founded only in this specific treatment? Are you aware on any other?
So yeah, so when we found out, we thought that nobody had seen that before, and but actually when I gave presentations, first of all, the group of David Kaltkins had shown that there was an exchange of glucose and metabolites between the two optic nerves, and he had a PNAS paper on that came out last year. But also two groups did mention to me that they had observed a bilateral effect, but one of them was a company and they never reported it, they never published about it. So this is the data that they never advertised about. But there is now some evidence that, especially for intravitreal injection, there could be a bilateral diffusion when you target the ganglion cells. For subretinal, this has not been observed so far. But I think now when you design a clinical trial for gene therapy, it's better to have a group that is untreated, completely untreated. Initially, we thought it would be unethical after the phase 1b/2a because we saw an improvement in the phase 1b/2a. So we thought it would be unethical for patients not to benefit from treatment in one eye because the disease is leading to legal blindness. So we didn't want to put patients at risk of not being treated. But I guess given what happened with this bilateral effect, we found out that maybe this is what we should have done.
Yeah, you know, there was a paper by, I don't know if you know him, Chris Passaglia. He's an electrophysiologist. And he showed, at least in rats, that there are fibers going from one retina to the other retina. Yeah. It demonstrated fibers crossing, and also that you can measure ERG from one eye if you stimulate the other eye. So maybe it's going all the way from one retina to the other with some specific retinal fibers. This was shown in rats. I don't know what's happening in humans.
Yeah, it's possible. There is also a couple of publications showing that mitochondria can move from one cell to the other. This occurred, actually this is used in some cardiology treatments, and there is some evidence that mitochondria can move from one cell to the other. So we don't know exactly what happened, but clearly it's not a placebo effect. It's not.
I also want to ask, I mean, it's beautiful talk and exciting results. I want to ask if you think that the fact that you had an effect on the untreated eye, in the fact that you see in the, okay, I'm not so, all these medical terms are not, I'm not so competent in them, but the fact that you see the disease progressing from one eye and there's a delay, and and the other eye is also starting to catch up on the disease. Do you think that it's also the same mechanism that, let's say, that the symptoms of one eye transfer to the other eye through the same mechanism that the treatment is passing through one eye to the other?
So nobody has a good explanation for the for two things. One is why do these patients have normal vision for so many years, and then all of a sudden, over the course of a few weeks, they start vision in one eye and the second eye. And second question is why only the eye? Because this is a mitochondrial mutation that is in every cell in the body, and only the eye has this. Actually, there is a symptom called Leber plus, where you have a few things in the urinary tract, but very limited. One possibility, but it's totally speculative, is that as David Kaltkins has shown, there is an exchange of glucose and metabolites between the two eyes. So let's say that if one eye is losing vision, maybe the second eye is helping a little bit, and then the second eye also is losing vision. But why is the crisis starting in one eye first? In 25% of patients, it's both eyes at the same time. But in the others, it's over the course of several weeks.
Mm-hmm.
I have a question.
Thank you.
Thank you for a great talk and great results. I think it's very good news for the people. My question is, if you show some very good and great improvement in visual acuity, do you have any other information about everyday life, like contrast sensitivity, or how these people feel improvement in everyday life?
Yes, so we have data on the visual field, but because the fixation is not very good in these patients, the visual field is not reliable, at least at the enrollment in many patients. We have data from contrast sensitivity. Actually, in the initial report we had on Reflect and Rescue, we had an even more important change in the contrast sensitivity for visual acuity. There is a change also in the OCT imaging between the treated and so this is something that has been observed. These analyses are ongoing. And the most striking is the quality of life. We reported in the papers more than 90% of patients in the studies reported an improvement in quality of life, very significant, which is very unusual to see this level of improvement. And this was done using 6 different scales. testing. So it's published already, and this is from Reflect and Reverse. Very significant improvement. Some scales, the improvement is in the range of 60%, and some scales, the improvement is in the range of 90% improvement.
Thank you, very good. I think it's, if I compare to other clinical studies, it's really huge effect and huge progress for the people. I hope that it will be included in other studies, you know, in other cases of people with blindness, because this is a really big hope. So
yeah, so we know we have to convince the agencies. We are very optimistic in Europe. The FDA is very placebo thing, so we have to deal with that. But hopefully we will. We know that patients want it, so this is something very important for patients. The next steps is other mitochondrial disease of the optic nerve. There are other mutations that could be targeted. So this is what we want to do now.
Joseph, one more question. So if you are giving mRNA, and this is translated into proteins, so how long do you think this will, the effect will be? It's not like replacing, it's not a gene replacement, it's a mRNA. So how long do you think will you have to do repeated injections?
No, because the DNA is in the cell, so the DNA is episomal, but the DNA is stable in the cell, so it's still making the mRNA continuously. Yeah, it's not like a mRNA treatment like we have now for vaccines. It's it's episomal, but it's continuous expression.
Okay. Okay. Any more questions? Okay, thank you very much. This was really fascinating, Jose. And we are looking forward to see more results and more genes to be supported. So let's thank the speaker.
Thank you very much. Nice to see you all. Thank you.
bye-bye.
Later.
We hope to see you in Israel face to face, maybe.
With pleasure. Thank you.
Bye bye. Thank you.
Thank you.